Hi everyone, welcome to our week 3 lecture recordings. So this week we have 2 chapter readings again. So we have chapter 4 and chapter 5. So this lecture is going to be just about chapter 4, which is all about research in psychology and the different research designs and considerations that we have to make when we're doing research. And then our chapter 5 lecture recording is going to be posted in a different link, and that'll be all about assessment, classification, and diagnosis. All right, so just a reminder, here are some of the things we reviewed last week when we read chapter 2 and chapter 3. So we learned different theoretical models. So we talked about the diathesis stress model, different transactional models, and different systems approach models. We also spent most of last week talking about the developmental psychopathology framework. So we talked about things like typical and atypical development, continuities and discontinuities. We talked about the concept of multifinality and equifinality, risk factors and protective factors, reciprocal and transactional processes. We talked about both biology and the environment interacting with one another, and the importance of studying multiple levels of analysis. We also talked about mediators and moderators, and then we talked about developmental timing, so things like sensitive periods and critical periods. And then we also, at the very end of that first recording, we spent some time applying the developmental psychopathology perspective to a case conceptualization. So we talked about different types of risk factors, whether they're individual level or contextual level, and that included things like phenotype, genetic markers, cultural issues, different aspects within the parent-child relationship, so things like that. And then we also talked about protective factors, also at the individual and contextual level. And then our second recording was all about the biological context and the environmental context. So our lecture was pretty short, and it was mostly intended to be a brief high-level review with the expectation that you would read most of that in your chapter reading. So we talked about the biological context, and we touched on brain development and the nervous system. We talked about genes and the importance of polygenic risk markers versus single gene inheritance. We also spent some time talking about learning context, so things like classical conditioning, operant conditioning, observational learning. And then we also talked about some of the cognitive aspects, so things like how our thoughts impact what we do and how we feel. And so we talked about cognitive distortions and cognitive schemas. And then finally, we ended on the sociocultural context. So we talked about different levels of the environment, whether that's family, peer, your community, or more societal implications for the onset of psychopathology in youth. So this lecture, again, is only going to be focused on chapter 4, which is all about research. So we'll talk about the scientific method, considerations we need to make when we're selecting measures for our research studies. We'll talk about research design, so the different types of designs there are, and the lengths of the different research designs that we'll cover. And then we'll also touch on ethical issues in research. So this graphic comes from your book, and these are just some examples of things that we might ask in terms of developmental psychopathology research. So in the center, here's our broad domain of developmental psychopathology research. And in this field, we might be asking things like, how common are disorders in youth? So what's the prevalence? Do these disorders change over time? So what does continuity and discontinuity of certain mental health symptoms look like in children and adolescents? What are the underlying causes? So what's the etiology, and what are the mechanisms for abnormality? So here we might be looking at risks and the etiological context, whether that's genes and the phenotype or more contextual level factors. Similarly, things like what risks and protective factors are implicated in psychopathology? What are the demographic factors associated with specific disorders? How do we treat disorders? How do we prevent them from even occurring? And what is the nature of the disorders that we believe currently exist, or at least what the construct is supposed to represent? So in the field of psychology, when we think back to the historical movements of the field, we always start with Sigmund Freud's analytic theory. And one of the major criticisms of his theories were that they were not testable. These were beliefs that Freud had based on his own observations and experiences and judgments as a clinician. And so when we moved to the second wave in the field, which was behaviorism, there was a shift into focusing on observable, concrete behaviors. And that's when we ended up, developing these principles of classical conditioning, operant conditioning, and observational learning. So with this shift to a more behaviorist approach, we really started to borrow from medicine and other hard STEM disciplines, if you will, in terms of testing testing hypotheses and assessing observable behaviors. And so the scientific method was one way of kind of allowing us to move into this behaviorist orientation. So here, when we talk about the scientific method, you've probably learned it in elementary school in like a science class. This is the broad scientific approach to research. So we always start with some knowledge base or some understanding of your particular area in the field of psychology. So for instance, if I am someone who studies parent-child relationships, attachment, and emotion socialization, I have some broad understanding of this particular content area. And in reading and learning about what's out there in the literature, I will start to identify certain gaps in what we currently know. And these gaps can be either methodological gaps. So for example, we might say, we have this evidence that this is related to this, but it's always tested using this one self-report measure, and really using an observational coding scheme that's been validated and is really robust in its measurement would be a more appropriate thing to use when we're testing these relationships. And it looks like no one has ever done this, or very few people have done it, and there's a real reason that we should be focusing our attention on this. So that's a method gap. We might say there's a measure or way of conceptualizing something that is seldom used, and now we have this belief that we should use it, and so I will be the researcher that is proposing this belief. So that's a methodological gap. There can also be substantive knowledge gaps, which is more about conceptually there's a gap in the literature on this particular subdomain of what you're studying, or maybe there's a variable of interest that has a relationship to your other variables that's not often looked at, or maybe there's a lot of mixed research, and so you feel that studying it in this particular way is going to add to the knowledge base. So as a researcher, you have some sort of knowledge of the area you're working within, you're identifying a gap or problem, and you're starting to generate your own beliefs and your own hypotheses based on the research that you've read and your own experience conducting research. So we identify a gap and we start generating hypotheses. And when we're making hypotheses, these hypotheses need to be testable and falsifiable. So there's a null hypothesis, which is the prediction that there is no difference statistically between the two variables that are being observed. So we want to be able to reject the null and say our hypothesis is supported. And so remember, the purpose of the scientific method isn't to prove our hypotheses. We always stand on the other side. We're trying to disprove our hypotheses because we want to show that our hypothesis is not just by chance. It exists in this particular context, and it actually is not just a chance occurrence, but because of this particular paradigm that we're using or this particular manipulation that we're using. The other thing I'll add is we're never saying that our hypothesis is true or that our hypothesis is wrong. We're always saying that we're rejecting the null and that our hypothesis is or our data is in support of our hypothesis or not in support of our hypothesis. So once you've generated your hypotheses and your predictions about the relationship between these variables that you're studying, and you've identified that it makes sense with the literature, then you can go about designing your study. And so when we're designing our study, we need to be able to make sure that we can actually test the measures that we are interested in. And so these measures are really just observable ways of assessing these theoretical constructs that we believe exist in nature. So we have to be able to operationalize the variables or the constructs of interest. So operationalization is how a variable is defined and measured for a study in concrete terms. So the example I have here is if you're measuring stress is a really broad theoretical construct, and there's a lot of evidence for it in different ways of conceptualizing it. If you're looking at medicine and you're studying more biological constructs, or if you're in a more psychological discipline, you might be looking at stress as a self-reported measure from parents, for example. So here we need to be careful and sure that how we're measuring stress is theoretically in line with the type of stress we're talking about, and the actual measurement of it makes sense. So you might operationalize stress as a combination of heart rate and cortisol response. If you're measuring aggression, you might operationalize it as the frequency of shoving or pushing kids in an observational context. Or you might measure aggression based on a self-report measure of a teacher writing about her perceived behavior or her perceptions of how a child is acting in the school environment. So it's really important that we are operationalizing our variables and using measures that actually tap into the concrete variable that we're talking about. So in addition to operationalizing, we also have to define what are the variables of interest, what is the direction or at least the association that we think is what we're trying to say in this research design. So we're identifying our independent variable, which is our predictor variable. We're defining our dependent variable, which is the responding variable that's affected by the independent variable. And then I didn't add it here, but if we have moderators or mediators, we are identifying what those variables are. And again, remember, we're never just throwing variables in there for the sake of it. There has to be some sort of empirical reason to include these variables in our model. So if I'm studying, again, parent-child relationships, attachment, and anxiety symptoms, I might say that I think a parent's responses, so like their parenting behaviors, is an independent variable, and I think the child's anxiety symptoms is the dependent variable, and I think that the child's, IQ, for whatever reason, is going to impact that relationship. I shouldn't put that variable in there unless there's some research around, IQ is either associated with the anxiety symptoms, it's associated with the parenting behaviors. There has to be some reason to throw in that particular measure. So we're never just throwing things in just for the sake of it. So next, we're going to select our research participants. So who do we want to participate in this sample? And remember that these are participants that are thought to represent a sample of a population. So this is a little bit, it's a little bit more complicated, and when you take statistics, you might learn this. But our study is kind of like a small simulation of something we think happens in nature in the population. So if we are interested in the general population of adults or children with mental health problems ever to exist, our study cannot capture all those people ever. So we are selecting a subsample of people, or a select number of people that we believe represents this larger population of people with mental health problems. So again, we're selecting a representative sample of the population of interest. There are different ways of selecting your participants in your study. Random selection is often the best way to achieve representativeness. So again, if our goal is to be able to kind of draw conclusions from our study, to be able to extrapolate it to the general population, we want our study participants to be representative of the general population. So if we are randomly selecting people, that increases our representativeness of the population rather than being pigeonholed and picking very specific people with very particular characteristics that are not actually representative of the general population. But there are problems with selection. It's not always the case that you can randomly select. And there are many different reasons, whether it's practical reasons, whether it's feasible, and whether that's actually what your study of interest is about. So, for example, if we're looking at kids who have suicidal ideation and really interested in conducting a suicide research study, most likely your population of interest will be people in clinics who are receiving residential treatment, they're in hospitals, or those might be the people that you have more access to because there's a higher concentration of people with suicidal ideation in these places. Versus if you're recruiting from the community, that actually is a little bit more representative, but you might have a harder time getting the numbers up for the number of people in your study, all of them who are endorsing suicidal ideation. So sometimes it's not feasible to get randomly select people, and sometimes it's not feasible based on the location you're at, the people you're recruiting, and different things like that. So just know that we want to be careful about how we are selecting our participants, but it's not a clean process. It's not always easy. And a lot of times we are having to grapple with the risks or the drawbacks of some of the decisions that we're making. So in addition to operationalizing and identifying your variables, selecting your research participants, you also need to select the measures. And so this goes back to the whole point of operationalizing our variables. So once we have a clear understanding of the construct of interest and how we want to measure it, we then need to select measures that are capturing the particular thing we're interested in and identifying measures that are strong in terms of their psychometric properties. So we want measures that have strong reliability, strong validity. There's evidence that supports the use of this particular measure in this population that we're interested in. So, for example, if there is a measure that has only been developed for adults, let's say 65 years old and above, and that's the only evidence base there is for this particular measure, it's never been used with kids or adolescents, so let's say 17 and below, we should not be selecting a measure that has never been used with children. Or if you are going to, then there has to be some rationale, and we might first need to conduct a study that shows, hey, this is a particular measure that is valid in this population. So that's not to say you can never use it, but if there are better measures that have been validated and are reliable in your population of interest, you should typically go with those measures. But if your research purpose, right, your study aim is to validate a measure in a particular population, that's a different story. But in general, let's say you just are picking measures and it's not a validation study, you just have an interest in, let's say, assessing parenting behaviors, you're not going to use something that is not validated in this population. And so the other thing we need to think about is, again, does it have strong psychometric properties? How closely does it measure the construct of interest? The third thing we're also needing to think about, especially with working with children and adolescents and families, is how are we going to collect the data? Are we selecting it from observation? So are we doing like a naturalistic thing? Are we going to a school and watching aggressive interactions between peers and the subjects? Are we going to be recruiting them to come into the lab, and are we putting them through a conflict task, and we're going to be coding and rating that based on some coding scheme? Are we administering questionnaires, and is the questionnaire going to be administered to the child, the parent, and the teacher? So those are different things we need to be thinking about. And then finally, once we have designed our study, we've gone through all the things we talked about in the previous slide, then we're ready to collect the data. And so that might take some time. So we go through the data collection process. And then once you have your data, then we move into analyzing the data and interpreting our results. So you will clean up your data, you will start analyzing it, and then we will interpret the results using some sort of statistical significance criterion. So remember, we talked about null hypothesis, and that's the hypothesis that there is no statistical difference that exists between the observed measures that we are predicting have a relationship. And so we call this null hypothesis significance testing, where typically we use some sort of significance threshold. So it's usually a p-value of 0.05 or less. That usually is like our criterion. And then when we run our analyses, we look at whether or not that p-value is below the 0.05 threshold. If it's below the 0.05 threshold, then we can reject the null, and we would say that our hypothesis was supported. So the p-value is the probability of getting our result if the null hypothesis is true. So remember, we always want to be able to show that it's not just a by chance occurrence, that there's some sort of variance being explained by our particular model. The other thing to note is that statistical significance, so that's like the p-value threshold, is not the same as clinical significance. So when we think about statistical significance, we're saying, is this difference real? Is there a statistical difference between the two groups that we're observing? But clinical significance is referring to, does this actually matter? And so that's a whole different story, but just note that statistical significance does not necessarily equate to clinical significance. So just keep that in mind. And then finally, once you've run all your analyses, you've made sense of them in terms of what your hypothesis was, you have set your criterion threshold, and you're looking at whether your data supports or is not in support of your hypothesis, then you're ready to write it up and report your results. And so the one takeaway of reporting results is just that it's on the researcher to cohesively kind of summarize the results in the context of the literature, in the context of their hypotheses. And when we say contextualize it and put it in the context of the literature and the research in the field, that also means talking about the strengths of the particular study and talking about the limitations. So if there are any threats to validity or reliability, it's our job to mention that, because this serves as sort of a groundwork for other researchers who might be researching something similar. They now have this understanding that here are the limitations of our studies. Perhaps another researcher could do this study and address one of those limitations. So it's on us to be able to communicate both the strengths and the limitations of our study. Because again, in the scientific field, it's not just about proving that things are right. We always want to be able to falsify. And so it's important that we are making clear our understanding of what the limitations of our research are. So we won't spend too much time talking about the specific types of measurement, because we'll cover that next week, or not next week, but in the next lecture. But this kind of goes back to the original slide about selecting measures that you want to use in your research study. So I talked about how there can be different informants, but remember, there are also different types of measures. So you might have direct observation, you might use standardized tests, you might use some sort of biological or physiological test, self-rating scales, informant reports, you might use genetic methods. So depending on your research question, there are many different types of measures you might be interested in employing. Another thing I referred to when we were on that measure slide in terms of selecting an appropriate measure for your research study, I made a comment that you want your measures to have strong psychometric properties. So we want our measures to be valid and reliable. So when we're talking about validity, we're asking, does this test or method that we're using measure what we want it to measure for a particular population? So remember, we talked about operationalizing our variables in really concrete, measurable terms. And I mentioned that there are different ways of conceptualizing different constructs. So I gave the example of stress. If you are in a science or a medical perspective or discipline, you might have a different way of measuring stress versus, say, someone who is a parenting researcher who is specifically measuring stress in the context of their parenting role. And it might be more related to their sense of self, right? So those are different ways of conceptualizing stress in different domains. So we want to select measures that are actually tapping into the construct of interest and are measuring what we want in this particular population. So measure should be an accurate indicator of the attribute of interest. And it's on an earlier slide, I didn't really touch on it, but just know that it's often really hard in terms of psychological constructs to get at exactly what we want to measure. So we already go into research knowing that there's, these are all contrived measures and theoretical constructs that we think we're measuring, but we can never actually say for certain that we're measuring exactly what it is in the population or in nature. So with that understanding in mind, that signals to us that the onus is on us as researchers to really select measures that have strong psychometric properties, given that we're already uncertain about the actual truth of whether or not we're measuring the theoretical construct of interest. There are many different types of validity or subtypes of validity. So I gave a couple or a few actually on this slide. So we can have internal validity, we can have external validity, we can have face validity, content validity, concurrent, predictive, or construct validity. There are several more, but I think these are the ones that you tend to hear most often in research. So we talk a lot about internal validity, which is, can I be confident that my independent variable is causing the changes I see in my dependent variable? And so when we address threats to validity, one way we can do that, or addressing threats to internal validity, I should say, one way we combat that is through random assignment. When we talk about external validity, we're concerned with whether or not our research findings generalize to real-world phenomena. Just a known risk and drawback is that when we increase our internal validity, which is our confidence that our independent variable is causing changes in our dependent variable, which we typically try to do in a really constrained laboratory environment where we use random assignment and a whole bunch of other methods, we know that we're also decreasing external validity, because the more tight a lab environment is, the more controlled our design is, the less generalizable it is to real world phenomena. We also talk about face validity. So does it seem like it's valid? We talk about content validity. So does it cover the appropriate content? We talk about concurrent or convergent validity. And so that's asking, do the results align with other measures of the same construct? So an example of that is, if I'm someone who is developing a new parenting measure, I want to know, does my measure have concurrent validity? So is it predicting or is it associated with the same things that other established parenting measures are known to be assessing or associated with? So if I have this novel parenting measure, I'm creating, or I'm interested in a psychometric paper, a validation paper of this new measure, one of the things I'm going to be doing is assessing whether it has any correlation with I can't remember at the top of my head, but the parental bonding inventory, the multi-dimensional scale of parenting, different parenting measures that I know exist and are well established in this research area. I'm going to want to make sure my novel parenting measure has some sort of correlation with. So that's what we're thinking about with concurrent validity. Predictive validity, we care about, does this measure predict relevant outcomes? And construct validity, we're asking, does this test measure the correct construct? So this is just an example, so putting into context what some of these words mean. So if we're thinking of convergent validity, we might use this example. So a self-report test of depression is highly correlated with an observational measure of depression. So that's kind of the example I gave with the parenting measure. So this is two different ways of assessing depression, and we're saying they have high correlation, which means they have high convergent validity. Matt's supervisor thinks his test of anxiety is actually probably measuring depression. So we think this has construct validity. It's assessing what we think it's assessing. Predictive validity. The SAT is highly correlated with university academic performance. So we believe that taking the SAT score taken in high school is associated or predictive of later performance on performance in college. So that is predictive validity. I will throw in a plug here that one of these questions will be on the first exam, so I would take a look at this slide when you're studying. All right, so we talked a bit about validity. Now we'll talk about reliability. So again, remember we want measures that are both valid and reliable. So when we're talking about reliability, we are referring to the consistency of a test or method. So we want observations to be consistent, or we want to be able to believe that if we were to administer the same measure again under the same circumstances, we would receive, or the participant would yield the same exact or similar enough results as this current time of testing. So again, there are different types of reliability. I only have a few here. So we have test-retest reliability, alternate form reliability, item reliability, inter-rater or inter-observer reliability, and then a whole bunch of other types of reliability. So just like how we talked about there are threats to validity, there are also threats to reliability. And one way of combating that is through standardization. So that helps us make sure that the administration piece is consistent across testing, across people, across time. A key word to know about is observer blindness, which is that raters do not know the hypotheses being tested. So this is important when we are administering measures that, let's say, depend on a rater to code some sort of behavior. So if I'm using a parenting paradigm and I'm having a parent and a teenager have a conversation and they're being recorded in the lab, we're videotaping it, we have audio tape, and I have a research assistant later watch that tape and use a behavioral coding scheme, I want that rater to be blind to the condition. I don't want them to know what the hypothesis is that is being tested. If this dyad, this parent-teen dyad is, let's say, in an experimental condition, so they received some sort of intervention, I don't want the rater who's coding my behavior scheme to know what condition they're in. I don't want them to know that they are a treatment person. And that's because we don't want to influence the rater's beliefs about what they're seeing in terms of the coding scheme. So observer blindness is something we can strive for to help combat threats to reliability. So here are a couple ways of thinking about the different types of reliability that were on the previous slide. So when we're talking about test-retest reliability, we are saying that given at time 1 and given at time 2, that participant performs relatively similarly on both occasions. So we are talking about reliability or consistency across testing administrations. Alternate form reliability is talking about two different forms or versions of a test. So we want whether or not people, let's say in this class, are given version A of an exam or version B of the exam, which is theoretically supposed to measure the same thing. We want those things to perform similarly across time. Internal reliability is less about the actual test method or the test construct, but rather how do people perform on it. So is this person taking the test internally reliable? So are they responding similarly across this particular measure? So are they responding to all the questions about stress and depression consistently throughout the exam, or are they kind of inconsistently, randomly responding a certain way? And then the last one is inter-rater reliability. So this is kind of similar to the observer blindness example I gave of having a rater code parenting behavior. So we are saying, so I gave the example of one person being a rater, but let's say you have two people rating some sort of coding scheme, which is often the case because it's usually really laborious and a lot of times you want to split the work between raters. That can help logistically, but if we do that, we need to make sure that these two raters don't drift apart. We want them to be really close in terms of their coding of this particular measure. So that's inter-rater reliability. Are these two people consistent in their ratings? So something can be reliable and valid. It can be valid only but not reliable, can be reliable but not valid. So this is, this graph I'm going to show you, or these images I'll show you, is something I often see presented when trying to help people understand the difference between the two. So in this example, we have a test that is reliable but not valid. So we see that they're consistently kind of clustering in this top left-hand corner, but it's not valid in the sense that we want it to be in the center. Like if this were a measure, we would say the most accurate way of, or most valid way of conceptualizing something would be in the center. In this next example, we have something that is reliable but not valid. Again, so similar. It's consistently falling in this bottom right corner, but it's not valid. It's not hitting in the center. The third example, we have something that's both reliable and valid, so it's consistently clustering together and it's spot on the bullseye. It's valid. And then on our other side, we have the last bullseye thing. We have something that's neither reliable nor valid. So there's lack of consistency, so all the red marks are spread out, and they're not valid. They're not clustering in the center, which would be the most accurate or valid representation. So we can have any constellation of reliability or validity, and our aim is always, when we use measures, to pick the ones that have the highest or strongest reliability and validity. So next, we'll talk about research designs. So up until this point, we've kind of talked about the scientific process just broadly, and we now have also covered what are the considerations we are making when we're selecting measures to use in our research projects. Now we'll talk about just the different types of research designs that are out there. So your book has a section on distinguishing between descriptive, non-experimental, and experimental research. So descriptive research aims to portray a phenomenon. Non-experimental research might involve some sort of statistical or other type of analysis. And experimental research comes the closest to establishing some sort of cause and effect relationship, because you're assessing, there's some sort of manipulation involved, which is why it's experimental. And it does also often contain statistical analyses in order to test whether there's that cause-effect relationship. In the experimental domain, you can have randomized studies. So these are the ones where you have some sort of manipulation of a variable to assess the effect of it. And then we can also have quasi-experimental, which includes manipulation of a variable, but participants are not necessarily randomly assigned to the manipulation. So randomized, the one above, you are manipulating a variable and you're randomizing participants. And then quasi-experimental is you have manipulation of a variable, but you're not randomly assigning people to a condition. So this is not an exhaustive list of the different types of research designs that are out there to exist, but your book covers, I think, 4. So the first one we'll talk about are case studies. So case studies are typically descriptive and non-experimental. So you're not typically implementing some sort of manipulation, although you might. Typically what a case study is for is to look at or to focus on the history of one unique case or individual. And a lot of times the rationale for using a case study is because this individual might have a really rare mental health problem, or there's some sort of reason that it's important to study this one case or individual. Again, we talked about threats to reliability and validity just in terms of measures, but also we can think about how this relates to our findings as a whole. So while case studies can be really rich and informative to help us understand kind of contextually what something might look like, the downside is that our findings are not necessarily very generalizable because they are so specific to this one unique individual. So the upside, though, is that it provides us with rich information and it helps us at least bridge the gap between some sort of clinical understanding of some phenomena in practice and bridge that with research. So I give a couple examples here. These are two papers that I know exist. This one down here on the left, on the bottom left, is CBT for an adolescent female presenting with misophonia, a case example. So this is actually a paper that I looked at when I was trying to treatment plan and prep for a client I was seeing last year who had misophonia. And interestingly, misophonia is not in the DSM, so it's not formally recognized as a mental health problem, in the DSM at least. And so because of that, there's really not as much research out there. And this particular client that I was seeing also had autism. And so there were kind of these unique circumstances of this particular client, and there wasn't much research out there that I could find to help me in trying to develop my case conceptualization or my treatment plan. And so this is one paper I came across that was exactly what it says. It's a case example, and this was informative, albeit not super generalizable, because it's really specific to this one female adolescent. My client was an adult, very different circumstances, but it was nonetheless really informative for helping to bridge some of the clinical applications that were discussed in this paper and some research applications in other studies I had seen. And I was able to kind of use both of that to help at least give some sort of insight into what I might be doing with one of my clients. So this is an example from the previous slide. It says case studies are typically done with something that's rare or there's not a lot of research on it. So that was this example. The one on the right, so enhancing capacity for optimal social and personality function through the mediational intervention for sensitizing caregivers, a case illustration. So this is yet another case study. We're calling it a case illustration here. And this is my advisor, so I know that the reason that this is a case study is, again, for kind of this rarity case. So this was kind of the first application of an intervention that had not yet been tested. So this was kind of a precursor to really formal trials of this intervention. And so here's another reason why you might want to even conduct a case study to begin with. So we also have correlational studies, and these are often non-experimental because we are typically examining the relationship between two variables without manipulating either variable. So in terms of ethics, the reason why we might want to look at a correlational study is because sometimes it's not ethical to subject a participant to some sort of manipulation that can be harmful. And so in this case, we might just be looking at the association between things that exist in nature as they are without any type of manipulation. So that's one strength of using correlational studies. Some of the drawbacks, though, is that because we're not introducing any manipulation, we really can't speak to the directionality or the strength of association. So because we're not manipulating a variable, we can't really speak to whether there's a causal relationship between these variables because we haven't experimentally messed with the temporal timing of the variables. A lot of times we use the Pearson's correlation coefficient to assess the correlation between two variables, and that has a range of -1 to 1. So when we talk about positive coefficients, what we're saying is as one variable goes up, the other variable also goes up. So for example, if I have a correlation coefficient of 0.85, so 0.85, that tells me that there is a positive relationship between the two variables, and as one variable goes up, so does the other. So for instance, if I am looking at maternal sensitivity and child's attachment security, and they have a correlation coefficient of 0.85, what that tells me is that just generally, as maternal sensitivity goes up, so too does attachment security in the child. In contrast, if we have a negative coefficient, what that tells us is that as one variable goes up, the other one goes down. So if we have a negative, 0.48 correlation between marital discord and attachment security, as marital discord goes up, child attachment security goes down, and the strength of the association is 0.48. So the strength or the magnitude is indicated by the coefficient's absolute value. So in the two examples I gave, I had a 0.85 coefficient and a 0.48. So I gave the example of a positive 0.85 correlation. That's a stronger magnitude. And then the negative coefficient was negative 0.48. Right? We look at the absolute value. So 0.48 is not as strong as a 0.85 correlation coefficient. So regardless of the positive or negative sign, we're looking at the absolute value to tell us about the strength or magnitude of that association. So I already said this on the other slide, but again, sometimes it's useful to use correlational studies when we just want to see first if a relationship exists before future research, or maybe we're not able to experimentally manipulate a variable. But then a weakness is that we can't really establish causality because we're just looking at associations. So there's this issue of directionality. We don't actually know that something is caused by another variable. So the takeaway here is that correlation does not equal causation. All we can say is that these two variables are associated with one another. And again, to illustrate this idea about correlation not being equal to causation, if we're looking at these two, the pink and the green variables, and we're saying these are associated with one another, we cannot necessarily say that this independent variable caused the dependent variable in a correlational design, because there could be some other third variable that actually explains it. And it's not actually that the pink goes or predicts or produces this green bubble. It might actually be that there's some other outside variable that actually is causing this relationship to exist. So we would call that a spurious correlation. So there might be some other reason that explains the association, and it's not actually the independent variable that necessarily causes it. Okay, and so we also have randomized experiments. So remember we talked about the experimental domain, and so randomized experiments tend to be the strongest method for inferring causal links. So we can have a clear hypothesis. So there's some independent variable that we're typically controlling some sort of manipulation of, and we have the dependent variable, which is the outcome of the manipulation. And we do this controlling to account for those confounds. So in the previous slide with the spurious correlation, I showed you that third variable that could account for the relationship between the independent and the dependent. Here in the randomized experimental design, we're actually controlling for that third variable. We're aware, and we're gonna account for that by really controlling the independent variable and any potential third variables. I talked about this already, but there's also random assignment. So if you have two conditions, let's say there's a treatment condition and a control condition, you are randomly assigning participants to receive one of those. There's different types of experimental design. So there's human laboratory studies, there's therapy outcome studies, and there's single case experimental designs. In the case of therapy outcome studies, we typically think about randomized clinical trials, or RCTs, and this typically tests whether a therapy reduces a problem in people who receive it. So with these sort of therapy outcome RCTs, we think about efficacy, which tells us how well a therapy works in this highly controlled environment with this really specific defined group of people. And we're also interested in effectiveness. How well does this therapy work in the real world? So remember, the more highly controlled a research design is, right, that enhances internal validity, but at the risk of reducing external validity. So this is sort of a similar thing here. We're saying with efficacy, how well does this work? Does this therapy work for these particular people in this research design? And the effectiveness piece is, well, how well does this therapy work in the real world for, let's say, other people with depression who are not defined in my research study who have depression? So again, we have some sort of treatment condition or intervention condition, and those are people that are typically getting the therapy modality, and then we are comparing it against a control group. So you can have either a simple control group, so typically they're not receiving treatment, so it's literally comparing a group who receives something, some sort of treatment, to a group that receives nothing. And so that tends to have some sort of ethical implications for research because we can't withhold treatment. So it makes more sense in other settings, maybe not so much therapy settings. There's also waitlist control group, which is basically saying one group is going to receive the intervention, the other group will not receive it, but then they will receive it in the end. So they just can't receive it right now at this point. And then we also have placebo control groups. So it's kind of the same interactions as the experimental group, but they're given some sort of inert substance. And then we also have treatment as usual control groups where there's already some sort of established program or treatment that this group is receiving, and that's what's going to be compared to the other group that's receiving a new treatment or modality that you're testing. So for example, if we are testing some sort of new therapy and we're testing it in an inpatient hospital setting where everyone is receiving treatment, that might be our treatment as usual control group. We can't take away their therapy, the type of service that they're receiving in the hospital. So they're going to be in our TAO group. They're going to continue receiving their treatment as usual. They're going to keep seeing the psychologist. They're going to keep going to their mandatory group counseling, whatever. Then there's going to be this other group that's going to receive the new therapy that we're testing. That's the purpose of this research trial. And so we're going to compare this experimental treatment to that established treatment as usual group. In addition to our therapy outcome studies, we also have single case experimental designs. And so that's when one individual or at least a small number of individuals are studied intensively. So we might manipulate one or a few participants, and this is sometimes called a time series design because the dependent variable is repeatedly assessed across time periods. And again, this is often used to evaluate the effects of clinical interventions. But while external validity, I think the problem with this is that external validity is not strong because of the low generalizability. So again, because we're focusing on this small number of individuals intensively, we can't necessarily extrapolate to the larger population. Within the single case experimental design, we can have either an AB-AB reversal design. So that's pretty common in clinical settings, and that's basically where an intervention is introduced and then it's withdrawn and then it's reinstated. And we can also have a multiple baseline design, and that's when an intervention is given to the same person but in different settings, or it's given to different individuals at different points in time. So just really quickly, here's an example of that AB reversal design. So you can see here we're assessing at baseline. That's when the intervention is introduced, and then we withdraw, and then we introduce, and then we withdraw. And then here's our multiple baseline design. So you can tell that there's multiple time points being assessed here. So I have this slide on animal studies because although in psychology and clinical psychology, we're typically using human subjects as our participants, as we know from the biology context lecture, biology also plays a role. And so because of that, we still have animal studies in certain fields of psychology. So not necessarily clinical psychology, but animal studies can be informative for later translation to human subjects research. So in animal studies, we can expose animals to situations in the lab that might be impossible or unethical to impose on humans. But again, there are also still ethical obligations that we have to fulfill to make sure that it's humane to animals that we are using as our subjects. And like I was just saying about the biological context, what we know is that most of our basic science research comes from animal research. So our understanding of the brain circuitry and neurochemistry started off with studying animals, right? So we were using like mice brains or what have you. and then we were able to study humans. Some limitations are that, again, we still have to consider ethical implications of studying on animals, and also this issue of generalizability. The brain circuitry of a mouse is not the same necessarily as a human, so we have to be able to understand the differences and know what can be extrapolated to the human population. We also have epidemiological studies, and so these studies really focus on the frequency and distribution of a particular disorder within a population. So here's where these are the studies that kind of yield those prevalence rates that we started to talk about during week 2, and incidence rates as well. All right, so we're getting near the end, and so we just talked about the different types of research designs. Now we can talk about these designs in terms of their time frame. So we can have cross-sectional studies in which we are assessing different groups at one time point, so it's kind of a snapshot. So we're making group comparisons typically between some group against another group. And so because there's this one time point that we're assessing the relationship, we still have this caveat of sometimes it's still not indicative of directionality. We can't really say with certainty that there's causality here. So what we often want to do if we want to increase our research rigor to be able to talk about causality is we like to implement longitudinal designs, which are really hard to do, really costly. If you're using human subjects, it can be really hard to retain the same people to study over 10 years, for example, if it's a 10-year longitudinal design. So cross-sectional studies are typically more convenient, less time intensive, easier to recruit and maintain participants, but the downside is that you're just comparing literally two groups at a different time, at the same time point. Whereas longitudinal, you're following certain people across a series of time points, and then you're assessing at those measures. You're assessing this particular measure at these different time points. And you can have either retrospective longitudinal design, prospective longitudinal, or accelerated longitudinal. So we mostly think of more quantitative research when we think about the example designs that we just covered, but you also have qualitative research in psychology, and so this is integrating non-numerical information. So that's what makes it qualitative and not quantitative. This is research that's not obtaining numbers or like empirical data, but rather some sort of qualitative indicator of whatever your construct of interest is. So it typically emphasizes real-world context. It's usually done through in-depth interviews. So you might have case studies where you're interviewing a particular client who's receiving a treatment, and that their testimonial or their statements is what is informative. That's the thing that we're interested in. So we typically obtain some sort of narrative data, we get interview data, we get life histories, so things like that. It's methodologically rigorous when we combine both qualitative and quantitative methods. So it's really valuable when we employ those things together, because generally the quantitative methods provide that more traditional data collection and hypothesis testing, so more of those numbers and statistical testing and modeling we're doing, whereas the qualitative tends to provide this more flexible, broad scope investigation. All right, so the last thing we'll cover are the ethical issues that we contend with in clinical psych research. So all scientific research must be ethical and legal. So you can look this up, but there is a history of abuse and unethical practices in this field when it comes to research in human subjects. And unfortunately, this history of abuse is actually what led to the enactment of having these ethical guidelines and ethical review boards that review research to ensure that things are ethical, procedures are not harmful to subjects, and continue to serve as kind of checks and balances for researchers who conduct human subjects research or animal research. So in different institutions, so University of Houston, for example, we have institutional review boards, or IRBs, which are basically ethical review boards who review all study procedures, review the entire research proposal, they look at our study aims, they look at our intended analyses, and they make sure that all of the things that we are proposing remain ethical and are not harmful to the subjects that we are going to be interacting with. So anytime you're making a proposed change to your study protocol, whether that's I want to add a new measure or I want to recruit the subgroup of X number of people because I want to ask these other questions, anything you do must be approved by your IRB before implementing any changes. And that serves as this balance to make sure that research remains ethical and we uphold this ethical integrity and responsibility. One of the things that the IRB will do is they review all your study procedures. They also review all of your measures, all of the forms you're proposing to use. And one of the things will include your consent forms. So they'll read your consent forms. Typically, universities have some sort of like boiler template that they want you to follow, and all confidentiality forms will make sure that you're touching on these key things because it's your responsibility as a researcher to obtain informed consent from your participants. So in the informed consent procedures, you are making sure that the potential participant understands the study. So they're typically given like the general study aim. They're assured of their right to confidentiality. So we make sure to talk about confidentiality and privacy. They're made aware that they have a right to refuse or withdraw at any time from the study. Typically, we try not to use deception, but if there's some particular reason that deception needs to be used, we try to have some sort of debriefing. And again, studies participants should be debriefed at the end of the study. So here are the things that we want to make sure we cover when we are creating our consent forms and when we are verbally obtaining informed consent from participants. Remember that informed consent refers to adults, so adults who are 18 and above who can provide consent. Assent is used for children and adolescents, and so typically we try to obtain assent from children and adolescents in research studies unless there's some reason that assent can be waived. So maybe there's kind of a question of competency or intellectual functioning that would not allow them to sign the assent form, that there are special circumstances in which we might waive that assent process. All right, so here is just quickly our little snapshot from the textbook. And then what is your weekly to-do for the week? We've just gone through chapter 4. Your next assignment is to read chapter 5 and review the chapter 5 recording. And then you should also start reviewing or making sure that you're kind of going back and reviewing your textbook, the lecture slides, the videos that I've recorded as you start preparing for exam 1, which is going to be next week. So in week 4, so it'll open September 9th, which is a Monday.